Hello, everybody. Sorry for this little delay. We had some technical issues sharing screen. My name is Alex, and it is my big pleasure to greet you all on behalf of the organizing team of AI Helps Ukraine and welcome you to the 7th talk of this online series. We are really happy to have with us today Professor Regina Barzilay to talk about machine learning for molecular modeling and drug discovery. We would like to, we know it's a little bit late, but we'd like to say a couple of words about why we are organizing this conference, this charity conference. So, Prof, Alexander.
Thank you, Alex. Hello, everyone. My name is Alexandra, and I'm a part of the organizing team. And I just want to tell you a bit more about our conference. AI Helps Ukraine is a charity conference in AI, where we bring together leading AI experts to present recent advances of their research. And in the same time, we're raising funds for humanitarian aid to support people in Ukraine. Unfortunately, the war is still ongoing, and many people are suffering right now in Ukraine. They live in the destroyed areas with destroyed infrastructure. They are in need of basic things like food, warm shelters, medical aid. And we're raising funds to support them with these things and also with medical devices, which help with wound healing. And right now, we've already collected funds for one device, and we need about $500 for another device, which will really help people who get injured with missiles. So please support us on our website. And many thanks to people who already supported us and to our partners, Mila, who kindly helps us with venue and logistics, and to Ukraine Medical Support and nonprofit organization, which helps us to process your donations and to buy and deliver all the supplies to Ukraine. And also many thanks to our corporate partners, Google Montreal and Evada Labs. And I give a floor to Alex to introduce our today's speaker.
All right. We're ready for that. So let me introduce the speaker. Professor Regina Barzilay is a distinguished professor for AI and health at the Jameel Clinic of the Massachusetts Institute of Technology, MIT, and part of the Computer Science and Artificial Intelligence Lab. She was first known in the machine learning field for her contributions to natural language processing, but in the last decade or so, she has mostly focused her research in the applications of AI for oncology, molecular modeling, drug discovery, and healthcare in general. For example, Regina was part of the team who discovered Halicin with deep learning, a new antibiotic compound that targets many deadly and powerful bacteria. In 2020, she received the AAAI Squirrel AI Award for Artificial Intelligence for the benefit of humanity, which was a recognition for her contributions to AI for good. And I personally look much forward to the research that will keep on coming out of her lab at the MIT. And it is a big honor for AI Helps Ukraine to have her with us today to talk about how to use AI to expand the reach of molecular models in the drug discovery space. Thanks for being here, Regina. The floor is yours.
Thank you. Thank you very much for inviting me. And I should say to everybody who's listening, I'm really sorry I couldn't figure out how to use the platform. So you can be an MIT professor and kind of have challenges with technical stuff. At any rate, I want to say that it's very special for me to speak in this event. What I'm showing you is a picture of me when I was, I think, 15 years old. So I was a student. I was a high school student in Kishinev. And Moldavia is very close to Ukraine, and part of my family comes from Ukraine. So I spent lots of summers in Kiev. So it's very personal for me what's going on there. I have a lot of family who now lives in the US who actually came from that part, and we all watch with horror everything that unravels there. I still, can't really believe that everything I remember from that time is kind of looks so very different right now. So thank you very much for organizers for creating this series. At any rate, let me just start talking about molecular modeling. So the way I created this talk, and it's like maybe updated version of the talk that I gave during keynote at ICML, is about how do we make sure that whatever we do in this molecular space is actually relevant for discovery, and we're really changing the way the drugs are discovered. So the work that I will describe today, includes contribution of many people. So you can see the pictures. So my faculty collaborator, Tommy Yakala, and our students and postdocs, and I would show the relevant pictures of the contributors during the talk. I tried as much as I could put references to the papers, because I would not go into great details about every individual paper, but there are references, so hopefully you can get more details if you're interested. So the way I'm thinking, so as Alex said, I switched to this field maybe in 2016 from NLP, and it's been quite a few years right now. And during this time, I sort of changed my perspective, because I learned a lot and failed many times. You know, what are the questions and how at least we here at MIT should be thinking about it. And I'm going to be talking about 3 aspects related to this modeling. One is problem formulation, another one generalization, and the third one is interpretability. So let me start with the problem formulation. So Alex, when he introduced me, he kindly mentioned the work that we have done at MIT on discovery of new antibiotics. But what I would argue here, that in some sense, you know, this discovery was more luck that really, you know, can be attributed to amazing discoveries coming from algorithms. So the first thing that I want to say, that if you're looking at people who really do discovery, drug discovery for real, like meaning, and who have a lot of biology background, most of work on antibiotics actually focuses on understanding the mechanism of action. So how do you put small molecule, for instance, that can interfere with some vital function of bacteria? And you see on the slide some of kind of known mechanism of actions that current antibiotics take. So for instance, you see here one of the antibiotics that binds to a vital protein and inhibits the vital function. So there are quite a number of them that are available. The problem with them, however, is that, as we know, bacteria develops resistance very fast, and many of this known pathway for interference are currently not very effective. So the Halicin, these drugs that Alex mentioned, happen to be extremely effective against multiple drug-resistant strains, like E. coli, C. difficile, and A. baumannii. Those particular pathogens are considered to be of crucial importance by WHO, World Health Organization, because there are multiple strains of these bacteria for which the current drugs are ineffective. So, but let's go back and understand how this discovery happened. So the first author of this work, his name is Jonathan Stokes. He's actually in Canada now. He's a professor in McMaster University. So he had these insights to take 2,500 molecules, which is really, really tiny. And I will tell you in a second why it worked, at least, you know, one of the reasons why it worked to use such a tiny set. And he screens them against E. coli. So you can see here wells where you put, you know, cells with, you know, impacted by E. coli, and put one molecule and you see whether it inhibits growth or not. So de facto what you get a molecule, and for each molecule, you know whether it had inhibitory activity against the parasite or not. And the reason, again, this particular data set sort of worked, because it was a very diverse library of molecules. It didn't only contain antibiotics, but it contained natural product. It had some cancer drugs, variety of different things. It's called broad library. So it had like really a nice type of diversity that enabled the model to learn. But so what we've done afterwards, we took zinc library, which contains, you know, hundreds of millions of molecules. I think we ran it on 200 million molecules, identify a few hundred, which Jonathan, in a very smart way, kind of identify how to filter the model's output. He selected molecules which happen to be non-toxic, which happen to be very effective according to our predictor, and also which didn't look like current antibiotics. He wanted to have something which is very different from the current known structures. So if you look at the Cell paper, it describes it in great details. But the point is that, you know, there was a lot of kind of human intervention there on the Jonathan side to make sure that whatever we're extracting is actually is likely to succeed. And also, even after he found this halicin, it took another many months for him to actually learn what was a mechanism of action, because this particular molecule, the reason it was so effective is because it had a new mechanism of action, so he had to elucidate it. And he actually afterwards told me that it's very lucky that we found it, within half a year. It may take even longer. So it really illustrates to you that while machine learning models can do something productive, there is a lot of human knowledge and intervention that is needed to really translate it into the drugs. And the question that, I am thinking about, how we can make this model much stronger, that we need to have less reliance on human intuition. And coming back to the question of formulation, we can think that there are multiple ways to think about how we can model molecular activity. So if you start on the very right, this is what I call mechanism agnostic phenotypical readout. That's what we did for halicin. You have molecule, you read some number, for instance, antibacterial activity, and you are learning the mapping between them. You know nothing about biological mechanism of action. You don't understand what it does to proteins. Again, you have a graph and a number. This is today a predominant formulation in molecular modeling. The second approach that you can take, you can actually say, you know what, maybe we have some knowledge about what's going on. Maybe we can at least say, which pathways were involved. Again, we're adding a tiny bit of knowledge and hope that it will help. But the final things that we ideally want to do is actually to understand what this small molecule is doing to the protein and how do they interact. And you can see that in terms of modeling, as we move from right to left, it becomes progressively harder. But let's start with a predominant formulation. So again, the idea behind this formulation comes from the traditional medicinal chemistry, where people identify that if you're looking at molecule, some of them have, for instance, toxicity, and the reason we know that, or have antibacterial properties, because they have particular functional group which correlate with activity. And previously to kind of machine learning models, chemists could identify what are these important groups. So the hope is, however, that there are many more groups or combination of groups that human experts know about. Therefore, what you can do, you can, take some set of molecules, screen them for desired activity, and then you have a set of graphs with a number. That number, again, represents some biological activity, and you would try to learn the mapping from the graph to this number. Again, as a computer scientist, we really love this formulation because we don't need to think about biology. We're just, again, into a graph world. And as we know, there are lots and lots of exciting developments in graph modeling, so we can just take them and apply it here. And again, there is a lot of simplification. Even though molecules live in 3D, we treat them just as 2D graphs. And also, it's fully mechanism agnostic, where you don't know how it happens. So again, previously people tried to do it to some extent. They use kind of a fixed representation of the molecule. They're called fingerprints. You can think it about as a vector that records different substructures that they identified as important. And from there, you can learn the property. The big advancement happened actually, I think it was 2016, the work by Devinot and his co-authors, when they realized that we actually can learn the representation directly from the molecule using graphs and predict the score. And he used, very, and by today, simple convolution algorithms, and there were a lot of developments. But what de facto all these algorithms do, however you decide to abstract your graph, is to map all these 2D graphs into some high-dimensional space where molecules with similar property, their vectors are close to each other. There is nothing surprising here. So the thing that actually did surprise me quite a bit is when I was preparing my ICML talk, I was really curious how well, the models that we developed at MIT in 2018 or 19, depending how you count, stacks against, the most high-performing algorithm in the space. And we selected something which is called TOX21, which is a standard benchmark. And I want to say, so that we save the time for question, if there are any, TOX21 as a benchmark change over the time. Now there is kind of a standard open source version of it. That's what we used here. And we selected algorithms which did very well on a variety of different benchmarks. And the student, Peter and Hannes, actually ran all of them on this benchmark. It has a few thousand molecules, I think 10,000 or something, which measures the toxicity of the molecules. And what was really surprising to me, that they tried these algorithms and some others. Those are the best numbers. So if you're looking at this ChemProp, again, an old model by 2022, the best numbers we got was 79.1. And if you're looking at the stronger alternatives, you actually get 80.7. So this is not what I expected, because again, given all the progress in this field, I was hoping to see really dramatic differences. And I would later tell you, why did it happen? Because we actually were able to discover why did it happen. But I would say that even at the time when I look at this performance, my question was, are we actually correctly modeling this task? Because the fact that we so much improved in the graph representation, it had such a minimal impact on the performance on this task, tells us something. Maybe we are not modeling what we should be modeling here. And with that thought, we can actually start thinking about another approach where we are going to add tiny bit of biology into this modeling. And in this case, we're just going to tell to the algorithm what are the proteins, the targets with which the small molecule interact, because most of the time when you get activity, there is one or several targets through which the effect is achieved. And the particular task that we looked at was, we started looking at it because NCATS, this is one of the institutes of National Institute of Health that works on the translation, drug translation, asked us to find synergetic combination of antivirals for COVID. We started doing it like in early spring 2020, when we became all locked in our houses. So here the idea was the following. So what you're trying to do, you're trying to take a drug, let's say remdesivir, as you can see in their particular experiments, it's experiments in the lab, and found that remdesivir killed 22.7% of the virus, however they measured it. They were trying to find other small molecules that are already approved as drugs, which could, when combined with this remdesivir, can be much more effective against virus. And here you can see on the slides a number of combinations that we found, and I want to say this is a work of Wengong Jin, and if you want to read it in more details, it's published in PNAS last year. But again, there is a formula which is called BLISS synergy. It's not like bliss, like blissful, it's somebody's last name. The idea here is that you are looking at a combination of molecules that their combined effect is more than the sum of its parts. And one way to think biologically how this combination works, it typically involves interaction with multiple targets, that they sort of have different mechanism of action. That's why they are more effective. Now, if people in NCATS who started working with us on it would have given us a lot of data for combinations, we would not have even thought of something different. We will just change our existing, graph convolution algorithm, make it optimize the BLISS formula and be done. The problem, however, was that the synergy data, when they give two molecules and identify whether it's synergetic combination, was very limited. We had less than 200 combination points. People always ask me why it was so bad, because again, at the beginning of the pandemic, you can only do this research in a very special facilities in terms of like antiviral development, and it was just not possible to get more. We did have enough data for individual molecules when we knew what happened to each molecule, whether molecule on its own is antiviral or not, but we didn't have data on the combinations. I mean, we only had this 200 points. So it was not very realistic to use this data to really learn the representation for combinations. So luckily, at the beginning of the pandemic, the biology community, which were way ahead of us in some ways, they already identify what are the different proteins that are really important for creating antiviral effect. And here you can see some examples that we extracted from the published literature that illustrate how and the roles of this protein in this antiviral activity. So what we did now, and it could be like really a minimal change to the model in some ways. You can say what we've done so far, we went from the molecule, we abstracted it into a vector and then predicted the antiviral activity. What we need to do now here is actually to introduce targets somehow. So we are going to go from the molecule, identify which targets which are relevant for COVID involved, and then from there predict antiviral activity. And of course, because we didn't know all the targets that are involved, it's a noisy information, kind of our representations that we are learning. In addition to targets, also kind of has a part of a vector that represents the structure of the molecule. So you can think about this kind of combined space that gives biochemical information. And you can train this representation using individual single molecules, because there is resources like Campbell, which shows to you activity of molecule for targets. So you can train on both on the individual molecular activity and targets in one go. And then afterwards, you can take this learned representation and fix them, and then just learn the Bliss synergy on this very small data set. And what we've demonstrated that using this target information, even with this tiny combination data, we already can identify synergetic combination with reasonable AUC. And we showed in the paper that having access to targets does help. But at the same time, this whole question gives us a pause here, because the way, and I'm going back now to the previous slide, the way I'm thinking here about the targets and about these proteins, it's really one hot encoding. You don't really know that some of these proteins are close to each other in terms of structure, others as distinct. You just kind of, it's like a symbol. And it really doesn't make sense. In ideal case, we won't go to this representation when we're actually understanding what exactly is a small molecule doing in relation to all the proteins it encounters. And of course, luckily, we need to mention this groundbreaking result from AlphaFold from DeepMind, which solved one of the open problems in biology, which is predicting the structure from the sequence. But what also happened, which is maybe not, creates as much publicity, but really important, that multiple groups, and I know that Mila is working on it, MIT, and many others, is actually trying not only to understand, to extract the structure, because for many relevant proteins, you already have correct crystal structure, but actually understand and predict how this protein interacts with each other, to understand the docking, which protein would connect together and have an effect, which will not, and also to understand how the proteins interact with the small molecules. So this is what we've been doing, and you can see some of the students below and postdoc who worked on this topic. And let me just illustrate one example of the paper that it was really interesting, because I will start with Equibind, which was, we were very proud of the paper when I gave the talk in at the ICML, but since July, it's kind of now retired paper, because we have much stronger approach. But I would at any rate say what we're trying to do here. So you have a protein where the crystal structure. It can be either predicted structure or correct crystal structure. You have a 2D representation of the molecule, and you're trying to identify where exactly this molecule binds to the protein, in which pocket, and what will be the orientation of this molecule. So you're trying to predict also the 3D structure of the small molecule. And the paper that I'm alluding to is a paper that now in review, to iClear, correct? That actually uses a diffusion process to look at protein small molecule binding. And the interesting part of this paper is the fact that it's actually designed the diffusion process around all the degrees of freedom which are relevant for the docking itself, where you will first identify the pocket and position of the ligand relatively to protein. You also look at the orientation within the pocket, and finally, we compute torsion angles for the conformation. So this particular, I just want to illustrate very briefly how I really like this video. It shows the green one is the correct location of the molecule, and the red one is where the small molecule ends up. So you can be asking whether I selected one example where it really works. So I should say that one of the strongest features of DeepDoc that it actually has really strong performance. And again, for those of you who are not familiar, the performance here is evaluated by comparing the distance between the correct pose and the predicted pose. And if it is within 2 Armstrong, it's considered to be good enough. And here you can see that for DeepDoc, we are achieving like 36 when the both physics-based models and the other deep learning model, including our own Equibind, are in low 20s. So this was kind of a significant improvement in terms of performance. And now the question is, what can you do with it? And is it good enough to actually utilize it in biological applications? And I want to give you some example of these biological applications that we are trying to utilize this model and to see if they really help. So this is a part of my talk that we didn't publish and recently didn't publish because we're still actively working on it. So I cannot even 100% tell you where DeepDoc is helpful here, but I would just want to share with you where we are in terms of our thinking, where we want to take it. So the first area that I want to bring to your attention is an area which has very little deep learning or AI. This is an area of metabolomics. So whenever we're thinking about biology, most people think about like sequences and genes and other things, but there is this very interesting area which actually looks at how particular, what are the biological reactions that actually drive what the mechanism does. So what you can see here is the reactions that are related to the contraction of the muscle. So you can see here that there is ATP molecule, which is shown on the upper corner. Which enables muscle contraction. When it donates one of its phosphates, the result in this muscle action. Here you can see the sequence of reaction. So pretty much every single thing that our body does, there is a set of reactions that are happening in the body with these molecules called metabolites. And here you can see, for instance, glucose metabolism, again, as a sequence of reaction. And here it's actually, you can see the same ATP molecules corresponding to it. So there are lots of known metabolic pathway sequences of reaction. Not all of them are known, many are. But the question is, can we take all this reaction and start using them to really understand, and here I'm showing you some of these reactions, can we actually take this reaction and to understand what exactly small molecules are doing in our bodies? And even today, when we are looking at various drugs that are developed, they actually interact often with known metabolites. And here you can see in red the drugs, and in yellow you can see, in a yellow kind of corner, you can see the metabolites themselves. So understanding what they do can help us, can help the kind of the drug discovery not just be some kind of correlation mapping as we're sort of doing it today, but really truly understand how do these molecules interact with different tissue in our bodies, what kind of reactions are happening here. And I would say that the reactions that I showed you at the beginning, they're kind of generic reactions that happen everywhere in our body. But today, there is a lot of data, even for specific tissues. So you can really ask what this molecule does for this tissue, what it does to our liver versus what it does to our lungs. So this is all in these very big reactions. And you can, again, ask a question. If I have a molecule, let's say an antibiotic, and I have all my pathways with which of the pathways it's going to interact for bacteria, and is it going to inhibit some vital features of the bacteria. You can also use it for biological discovery. As I said earlier, we understand quite a bit about generic reactions in our body, but there are a lot of tissue-specific reactions which we don't know. It's very hard biologically to collect it, so can we do a better job? And finally, a lot of products are today generated through biosynthesis. So bacteria are used to produce chemicals. You can ask yourself a question. If I understand all the metabolic pathways of the bacteria, how I can interfere, let's say, to increase the production of certain material, or alternatively to change materials that it produces. All of it requires us to model these reactions and the concentration and the flux of these reactions. And just to mention, I don't want to go into great detail, but just to mention, we did something with antibiotics where we took the metabolic pathways of E. coli, we took the small molecules, and we trained to predict which of the small molecules can actually inhibit the vital function in bacteria. This is just very preliminary results. But what's interesting that using this approach, we can already do better, tiny bit better, let's say comparably, okay? Let's say comparably with approaches like approaches that, you know, were developed forever and, you know, are much more standard now. So I just really wanted to see in this experiment that we can at least match it. And I think there are so many things that can be done. And specifically, let me just tell you, so for full disclosure, what we didn't do here, in this particular case, we took the graph as it is. We didn't really model the new reaction. We took the existing embeddings. But the key question is, can you identify what these reactions are doing on the flight? And here you can see one example when you have an enzyme and you have a small molecule and a metabolite, and you can see one reaction where you kind of switches the direction of the NH3, one functional group. But we need to have a method for doing this kind of prediction on the fly in order to be able to ask, you know, is this, what would be the result of the reaction? Or if we know the outcome of the reaction, what were the ingredients, you know, what are the right concentrations? So we really need to be able to use something like DivDoc to help us solve this kind of questions. And so far, we're just working on the solution. Another area which I, again, strongly believe that approaches like DivDoc and others can help us solve is, you know, immunology. And one interesting thing that, you know, I discovered because I was studying, you know, immunology at the baby level, I should say, in the last year or two, because I have students who are really working on it. So we've been getting educated in the field. But one of the things that I'm sure we all know, that T cells that we have in our body are very, very specific. So by looking at what they bind, you can actually detect what kind of disease a patient may have, because every type of cancer has its own mutational signature. So conversely, you can look at T cell and say, if I know that this T cell actually binds to this epitope, which is a characteristic of lung cancer, I can, by looking at the T cells that the person has, actually say whether they are likely to have lung cancer or not. So the question that we ask here, and this is a collaboration with biologists at MIT, if you implant, you know, cancer into a mice, they have this very special mice. I would not comment on it because I don't truly understand, you know, how the one mice is different from another, but this is kind of informative type of mice. At any rate, what they've done with this mice, they implanted into it a lung cancer. And the question that we asked is how soon you can identify the T cells, which are characteristic to this lung cancer. What it means that you actually gonna take blood from the mice, extract, you know, the T cells, sequence them, and see which one of them bind to the epitope. And what this experimental result demonstrates that within a very short time, within 2 weeks, before you see any indication on the imaging of the lung cancer, you can already see the spikes in T-cell repertoires that correspond to the lung cancer. And now, again, you can take these epitopes and T-cells and predict whether they bind or not. And instead of doing assay, you can totally do it in silico. And the hope is that you may not only use it for diagnostics through blood, you can also utilize this capacity directly in designing of new therapies, which are now so extremely expensive because you need to do a lot of empirical testing that you can now with new binding approaches actually do it all in silico. So this is the type of questions that we're trying to address. I wish I would have told you that we've demonstrated the DeepDoc and all the other things that we developed here is making dramatic difference. I cannot yet say so, but I think we're getting in this direction. I'm really excited to see where it will bring us. But now I have another few minutes, so I just want to talk to you about the topic which I'm kind of carrying with me because I think it's a topic of a great significance, which very much relates to what I told you when I started my talk. We need to make sure that whatever we are producing is actually has relevance. And the first question that any chemist would ask you is, you develop a model, would it generalize to a new chemical space? And the reason they're asking it, because there are some chemical spaces, some subsets of the space that are very well investigated. And you really want to apply it to other parts of the space. But how we can really say whether we can generalize. And if you look at life sciences, there is a whole bunch of people who utilize different heuristics. Like, for instance, if you're doing biology, you would do batch split or you do scaffold split when you are a chemist. And again, what is scaffold split? You take all the molecules, think of them as graphs, and then you kind of cluster them based on their backbone so that you want kind of cluster of molecules that look one way being in training and the other one being in testing. And this is very different from what we do in natural language processing or computer vision, where you do random split. So the first thing to say that clearly, if you even use a heuristic method like scaffold splitting, you drop quite a bit in accuracy. So you go like in the, I think it's TOX 21 or something, you go from 78 to 54. It's like a vanilla model. But what I would show you is a method where you can drop even further, where you can find even a harder set where you get to the accuracy of 33. Oh, sorry, ROC AUC of 33. The idea here is to be able to automatically identify a subset of, in this case, images or molecule or whatever it is, which would be in some ways a minority, which would be hard to generalize to. So in this case, you can see samoyed and a bear, and you can see that samoyeds often appear near the grass and bears appear near the snow. So it will be so this association between the background and the label clearly would be picked up by a learner. So the cases where it flips, it's most likely we'll find them as hard cases. So you want to be able to identify this minority group automatically. And the algorithm that does is actually extremely simple. You split your set. You start with a kind of random split of your set, and you train the model on the training portion. And using this predictor, you're kind of learning how to move training and testing point around in such way that your generalization is the hardest. And you do it with a subject to constraints where you want to keep, roughly speaking, the same train test ratio and similar label distribution in training and the testing. So we tried this model on a variety of different data sets from like imaging and natural language, but I want to bring to your attention the result related to TOX 21. You remember that when I started talking earlier, I mentioned that for some reason, at the time it wasn't a known reason, but for some reason, all the known algorithms don't really do great on this TOX 21. And I was wondering, what's wrong with the data set or what's wrong with an algorithm? So we use this splitting method to identify this minority subset on which the model really poorly generalized. And then we look at those molecules and what's so different about them. In terms of like tiny motor similarity, they didn't look very distinct from the rest of the molecule in the training set. And what we found out that if you look at this data set, TOX21, we didn't use it for training, but also what you can see in documentation that can actually tell you what are kind of the mechanism of action to which the molecule achieved toxicity. And what you will find that this data set, which was really hard for the model, is the one where the relevant targets, the mechanism of actions were quite distinct from the rest. And also there was not enough of these targets in the whole training set. So in this case, you sort of start to understand that the reason you cannot generalize, you're generalizing, okay, in the space of molecule, but you are implicitly asking the algorithm to generalize in the space of targets where it never seen those targets, and they are significantly underrepresented. In some ways, doing this kind of splits one direct benefit that you can actually afterwards ask yourself a question: why exactly this data set is hard? And sometimes you can find explanations, sometimes you don't. And even if you don't find an explanation, but you still want to be able to do well on the subsets, you can use algorithms like DRO and others to kind of learn in a way where the majority that doesn't, kind of trumps the minority to get a good performance. So you can still, with this algorithm, achieve okay performance on the smaller subset. And what I want to say is that we actually looked at the mapping between this automatically induced hard set and the scaffold split, and we see that they really don't map well to each other, which gives you the idea that these heuristic splits are really not a good idea. And here I'm showing you an example of a paper that is just a random paper that comes from, I think, Nature Machine Intelligence, but you can find them everywhere. It's a paper that looks at, a data set and tries to, to do some prediction with the biological data. And in this paper, like in any paper, there will be a significant portion of the paper which is dedicated to data cleaning and how you remove noisy samples. And this is really funny that to this day, we are by hand doing this cleaning where the cleaner has a lot of leverage, removing hard examples. How do you decide what is noise and what is a hard example? Sometimes it's really, really fuzzy. And, what I hope as a community we're going to develop into the sets of tools that we can use to properly reason about generalization, where we have a clear criteria of what we want to generalize. And when we don't do this kind of heuristic on cleaning, because then we don't really know what will happen when we're applying it for real. So this is an area that I really greatly care about. And I really hope there will be a lot of interesting solutions moving forward. And the final thing that I want to say is to say about interpretability. There is a lot of research in our community, I'm talking about machine learning in general, was dedicated to interpretability. But, in the last few years, the more I look into it, the less convinced I'm becoming. And I should say that I also wrote a paper on interpretability like everybody else. So what do we think, like most of this rationalizing paper are thinking about it in the following way? Let's say I have a small molecule which have some toxic activity. And I'm going to say, okay, the reason this molecule is toxic is because it have this particular functional group. And we as human knows, chemists know that this is kind of a toxic force. So that's why the model did the right prediction. But the problem is that most of the time when we are doing this kind of predictions, we actually don't know what is the right answer. We don't really understand which part of the molecule drives the property. So first of all, humans cannot validate it. And second of all, the reason this molecule is toxic, it's not because of this group. The reason it is toxic because it actually interacts with other proteins, because it does something, not just because you have a particular functional group. And as we move forward, this is an example of another application where, again, you're trying to predict the future cancer risk. Again, humans cannot do it, machines can, but humans cannot validate it because they don't really understand what to look at. So how you can validate a prediction if you as an expert don't know the rationale. And I just want to bring to your attention, I have like 1 minute, papers that we just recently completed that will be published in Transaction of Machine Learning Research that looks at the model which do selective calibration, where you teaching the model which examples, which part of the space are staying well calibrated and which ones are not, so that your classifier kind of staying. And I would, unfortunately, I forgot to put the reference to the paper, but hopefully you can find it. But again, the idea is to accept the fact that we cannot understand, but at the same time, enable machine to know when it doesn't know, to be able to tell to humans, I don't know, so that we can really find a way to trust the confidence that machine gives us. And just to conclude, I want to say that it's not like I don't believe in interpretability, I do. But I think in order to really reason in biochemical space about interpretability, we need to have a lot of context that can mechanistically give us a rationales. And at this point, we are not quite there. And if we are not there, instead of using, this very simplistic rationales, we can use other tools that help us to deal with, to handle uncertainty of our predictors. So thank you very much. I think I am done. Let me stop sharing.
Yes, thank you very much, Regina. That was a very great talk. A lot of very interesting research and very recent there. So thank you very much. Yeah, we're almost at the hour. I don't know if you have a couple of minutes to answer some questions, or do you have to?
Oh, and thank you. Somebody put the references to the papers. Thank you very much.
Oh, yeah. There's one very concrete question by Yep Huang, who was asking about the name of the tissue-specific molecule data sets, the part where we're talking about like tissue-specific reactions.
I think there is just a link. So if you can email me, I will send you. It's publicly available data set. I think it comes from UCSD, from University of California, San Diego. So just send me. I will send you a link to it.
Perfect. So audience, take a note of that. I'm personally quite interested in all the last part about data splitting and how important that is. And I see there's a big tension between different things that we would like to achieve. Like we want to use as much data as possible so that we can generalize to many things, but at the same time we want to make sure that the progress we make is relevant for different tasks. So that's why we want to do a smart splitting depending on what we want to measure. And then there is like scientific reporting, and we want to be state of the art and so on. So how do you see the near future of machine learning for these kinds of applications in this regard? Like, are we going to really be able to do this cultural change and yeah, for the better, hopefully?
So the way I'm thinking, and since I've been for a while here in the field, I can see the progression. If you think about like the time that I was a graduate student, before I started, people were just doing rules, correct? There was no machine learning, there were just rules, and they built a system for NLP with the rules. At the time when I was a graduate student, there were already the first like decision trees and some models, and most of the PhD, what people were doing was actually designing features. And this was the whole PhD. You could write the whole PhD. How do you design the feature and what we call today representation? How would you design by hand representation? And then the next step that came was to actually learn the representation, correct? So we're really moving away in all stages from our, human intuition, because our intuition is very limited. But I think one area which we still are very much driven by our intuition is all this area of how we do splitting, how we identify biases and all the other things. And to me, if you look at the large body of work that community contributes, which is a great thing on fairness and stuff, many of these papers start by saying, we know what is a minority group. But the problem is, and that's what we see again and again and again, that we don't know. And this is the worst case, because somebody gets systematically discriminated because we don't know that they are unique in some way. And to me, it's not about human intuition. It's really about creating the tools that can detect the bias, which is algorithm specific, that can detect the shifts, that can reason about the uncertainty, these meta tools that I think community is kind of slowly moving into this understanding, but we are nowhere close where we should be. And I would, if I may use my few minutes, to whine, this learning to split paper, like the reviewer was asking, why do you need it? And my point is, the reason you need it because you are creating models which nobody is using, because the random split doesn't work. And I learned it on the personal example, being, like this is my office, and just across I can see Amgen. And I remember when we developed the first molecular model, I ran to people at Amgen and told me, use it instead of this very basic stuff you're using with handcrafted features and with your old decision tree. We can do much better. And we gave them the ChemProbe, and it didn't do better. And the reason, it was the first time that I realized that they actually doing temporal split, that they intentionally separating the test to be very different from training. Of course, you can achieve it with the right regularization and so on, but I was not even aware that that's what they're doing. And my point is that this random split that currently dominates the community, may be applicable to some applications, but a lot of this application really, this is not the right way to go. And as you said, Alex, that sometimes we can say, what is the right degree of generalization? And depending on the application, maybe somebody wants to stay within the same chemical space, and it's fine. But sometimes people really want to go far, and you need to have some, probabilistic measurements of this shift that you're trying to achieve when you're testing your model. And again, human intuition is really not the right way to go forward. Yeah. So I hope we'll see more shift, but for now you can go to Open Review and see what people wrote about the paper, and you would see it's very, very interesting that people really don't see a value of asking these questions.
Yeah, it takes a while to make these cultural changes, but I personally think it's very, very important research. Thanks for that. Fortunately, I see some recent data sets providing different splits to measure different things. And
this is the point, that you can do different splits, because to me, the problem when we're again thinking about a bias, it's not what we know. If you know that you're working on a medical data set, you can stratify it by race, age, and preconditions. This is easy, but think about this example that everybody gave. I remember that it was like kind of popular. We all use this example in our talks related to, what is it called? This actress, Catherine. Are you familiar with this example? Mm-hmm. So what is, Catherine Hathaway is an actress, correct? And there is Berkshire Hathaway, which is this hedge fund or something, some financial institution. The next thing that happened was that somebody found out years after it happened that each time when this actress was in the news with a new movie, the Berkshire Hathaway stocks went up, because the robotic trader was connecting the actress Hathaway to something which was totally unrelated to the Berkshire Hathaway. And it's Buffett's, I don't know, financial entity. And the worst part of it, that it was not discovered by machine learning person, it was just discovered by blogger based on the correlation. So the question is, how many of the systems that we deploy, that we are currently using, are making these very terrible mistakes, and we are not aware of them, and assume that it can be detected by human intuition is one of the most dangerous misconceptions.
Yeah. Thanks for the answer.
Yeah, we have one more question from Joshua. I just invited him on the stage.
Let's see if he can join
us.
Hello. Do you hear me?
Yes.
We can hear you.
Yeah.
Go ahead.
Hi. Thanks a lot for the great talk. I had a sort of difficult question that I'm asking myself. I wrote, have you thought about machine learning methods to discover the causal graph of reactions? And then how do we make this at scale? Because right now, as you said, we're like using the metabolics, the reaction graphs, and the gene regulatory networks and so on that biologists have found, but it's clearly just a piece of the real action.
So this is the bigger goal. This is a bigger goal, and I hope that's where we as a community are eventually gonna end up if we are successful. But at this point of my life, I'm spending more time thinking at much more basic kind of one-step approach. That, you know, what is the right way to think about the interaction and the reactions of, even if you're looking at one-step reaction between the enzyme and small molecule, what is the right representation of the enzyme? Because we know that it happens, the reaction itself is very local. We also, you know, we're really overemphasizing currently, again, due to the AlphaFold, utilizing the 3D structures, correct? And we're really working, building those representations. But what we see, and we hear it from others, that just simple embeddings currently can predict the reactions better. So there is something that is not, you know, kind of makes sense to me, because, you know, on one hand, I would believe that 3D should be able to predict these reactions better. That's not what we're seeing. So I'm just trying to do it on one step at a time, but eventually I do see the big causal graph of reactions as our ultimate goal.
Yeah, thank you. I guess our current 3D models are just not good enough, and the errors just, you know, are, you know, end up accumulating and being useless. Yeah.
So I don't know, you know, the question again, if I look at NLP, because I came from NLP, I don't know, you're sure if you were following the research in like all times, but the problem in NLP, you know, when I was a younger faculty and a grad student, was how do you do syntactic parsing? There was enormous resources that were were spent on creating, you know, the annotated trees, and, you know, all the top universities had groups working on syntactic parsing. But as a representation, and we believe that if you get the syntactic parsing, you're going to solve machine translation, information extraction, and all other areas. But you see that syntactic parsing is nowhere used in all the models, and they're still doing a fantastic job. So the question that I'm asking, I'm even afraid to verbalize it to this audience, but the question that I'm asking, would it be the case that we don't need 3D representations? Because the 3D representations are learned from sequences. So maybe we can make these sequences in such a way that we don't really have to use the 3D representation. And again, I think that AlphaFold is the greatest achievement, but maybe it's not what will make the difference in the end. And we just need to change our thinking, the way we start thinking differently about machine translation, correct? So I think we need to be open to different possibilities, yes, rather than merely one approach.
Absolutely. Thanks a lot for your answer and the talk.
Do you have a couple more minutes for some quick questions?
Yeah, go ahead.
Oh, perfect. Yeah, for sure. Go ahead.
Okay, so the next question is a bit technical, and Yep asks about the name of the tissue-specific molecular dataset. I guess it refers.
We already answered.
Yes.
Yeah.
Share, and I want to encourage maybe your. I think it's there is a lot of data. There are areas in which you don't have enough data on the metabolomics and reaction. The data is available. It's available on mice, on bacteria, and on humans. And there is data which is tissue specific and not tissue specific, general reactions like the one I showed you at the beginning, but there are a lot of specific reactions. And if I may spend some time talking, because I really want the community to get into this field, is there are a lot of interesting questions. Because if you look like at different types of bacteria, like E. coli, but different strains of E. coli, they actually have different graphs. I mean, 90% are preserved, but 10% are different. And you can determine what will be on the graph, at least to some extent, looking in the transcription, because it tells you which enzymes are present and which are not. So, you know, like when we're thinking about antibiotics, again, one of the big problems here that when we're selecting one that maybe works on all the known species, but we're not looking forward to see what's to come next. So maybe we can reformulate the question and ask ourselves, how do we create a molecule which will deal well with different strains that we haven't even seen, because we can imagine how the reactions would go, like just kind of looking at the transcriptomic or possible transcriptome of this bacteria. So there is really a way to utilize this information in a novel ways. The information is available, and I really hope that we will be not the only ones who is looking into it. And I'm happy to send to Alex and whoever emails me the full list of materials that we collect, that we identified in this field.
Thanks.
Thank you. Thank you so much. One more question from Maxim Koroblev. Thank you for the talk. Do you use DivDoc to design T-cell receptions de novo?
So this is, we're actually looking at the T-cell distribution, but one thing in T-cells is the following, that the best models that we currently have on everything we do in immunology are sequence-based. I think there is just one case where we are using structure is for immunogenicity. We couldn't really demonstrate that the models based on structure do better. Again, it could be the case that sequences are sufficient with the best models. It could be the case that we are not doing something right. But for the performance, I want to tell you how the T-cell binding, at least what we identified, that if you have enough information, if you have epitope, and you have enough training data for epitope, with what any model will do well. The key challenge is to be able to generalize to new unseen epitopes, because it's an open set, and that's where we still hoping that we will make structure models do the magic. We still don't have results for that. We're just working on it.
Thank you. One more question from me. I wanted to follow up about generalization. I wonder how do we measure this distance in the space? Do we look just at the graphs, or do we look at the chemical properties of the molecules?
So the way we did for at least this model, there are many other approaches. We kind of look what is the worst data set to generalize in terms of your loss function. So typically when we're talking about the loss, we're talking how to minimize the loss. Here you're saying, what is my subset on which my loss will be the highest? And in this case, you're not just looking at the molecule, the graph or whatever, but you're just looking at the graph and label together, because it's not the graph. That's what I was kind of trying to say, that it's not just the structure itself, that what scaffold approaches do, they separate them based on the graph. It's more about the relation between particular graph and the label that it got. So that's what we have done there. And there are a lot of questions that we're now experimenting, because I don't know the answer. Like this particular algorithm, the way it is designed, it actually looks at it's algorithm specific. That predictor is your algorithm. So you can get one split for one algorithm, say this is the hardest for this class of algorithm, and some other split for another one. So we have somebody actually in my class, undergraduate, he's trying to see what variation do you actually get. Is it the matter of your training data, or is it something about the algorithm? I don't know the answer. Another question, actually, for those of you who work on protein network and others, you know how fully corrupted are the existing training sets. So it's a natural way to try to kind of to disentangle them. But then the question is how much your prediction will change when you are moving to how this prediction will change when you are looking at a different representation. Let's say I represent protein using, the sequence, one algorithm for sequence versus another, is the set is going to be different. So we don't really know. So this is kind of things that we need to decide, is it a good thing or a bad thing? And I hope that it's not going to be the only algorithm. We will have other mechanism to solve it algorithmically, but at least you have some measure. You can compare the challenges of the data sets.
I see. Thank you so much.
Yeah. Okay. So if you guys have any questions, you can just email me. There is just one Regina Barzilay, and my email is there, so you can email me. I will be happy to answer. Okay. So thank you. Thank you very much for what you are doing, and I really enjoy giving this talk. Thank you.
Thank you. Thank you for the talk.
Have a nice day, and a lot for joining this. Bye-bye, and bye everyone from the audience as well.
Yeah.
We will put the link to donate in the chat once again, and you're very welcome to share with your friends and colleagues. Thanks everybody, and see you soon for the next talk, which is on It's this Thursday by David Ronick, Professor David Ronick, who will talk about machine learning and climate change.
Thank you, everyone. Bye-bye.